before we get there, I'd like to talk some more about TCA cycle, finish up our discussion of that and show you the mechanism. We noted that this cycle is quite complicated, and it's generally thought that the reason that cells retain it and originate it is because it clearly has adaptive value in evolution, and the basis of the adaptive value seems to be the fact that it serves as a metabolic hub, lots of in and out reactions that connect different parts of metabolism, one prominent part being amino acid metabolism, but also fat metabolism and other aspects of secondary metabolism that we won't be talking about. So we've got this cycle of 8 reactions. We talked about pyruvate dehydrogenase complex, which generates the acetyl-CoA at the very top of the slide. And that was the monster enzyme, you'll recall. They used 5 different cofactors. It was constructed as this gigantic spherical-like particle with lots of reaction centers on it with 3 different enzymes and the channeling of the substrate as it went through the various steps. So that's to generate acetyl-CoA. And then we mentioned last time that there's another enzyme in the cycle that is kind of just like that, and that is alpha-ketoglutarate dehydrogenase complex. So this complex and the pyruvate dehydrogenase complex, the mechanisms are essentially the same, and the only difference is that this guy's feeding in a 5-carbon compound and shooting out CO2, and PDC is shooting in a 3-carbon compound and putting out CO2. Otherwise, it's a matter of the substrate specificity. Otherwise, that whole bigness, the large particle, all the cofactors, those are all there for this complex right here. So we talked about some of these steps, and then we got to this oxidative decarboxylation here, and I noted that for isocitrate to be converted to alpha-ketoglutarate, and I noted that it's a carbon-carbon bond cleavage reaction. If you look at the pentose phosphate pathway, one of the early steps in the oxidative part of it is another oxidative decarboxylation. And those reactions will proceed, we'll be able to cleave a carbon-carbon bond without a positively charged electron sink to stabilize the carbanion, like the shift base in aldolase, or like the thiazolium ion, a thiazolium cation in thiamine pyrophosphate, TPP. Both of those are positively charged sinks that can function in resonance with a carbanion to stabilize the formation of the carbanion, which is the tough step of cleaving a carbon-carbon bond, right? So now we're cleaving a carbon-carbon bond here, and of course we're releasing CO2, which is very stable, and so thermodynamically it's very favorable. We know that this is actually one of the irreversible steps here. There's no reverse arrow going back, it's just going forward. And that's because CO2 is such a low energy molecule, and if you just look at the structures, right, to think about the delta G, or at least the delta G0 prime, one side of the reaction versus the other, is going to be favorable forward because this is so low energy. But that doesn't talk about the kinetics, right? The fact that it's thermodynamically favorable and has a strongly negative delta G0 prime to go from 3 to 4 here, to go through step 3, that doesn't mean that you've explained everything you need to explain with respect to how it works, because we still have this issue of how do you cleave it. Even no matter how favorable this is, if it takes a million years to get there because there's no good rate enhancement, then that's no good to the cell, right? So the distinction between thermodynamics and kinetics. Is that clear to everybody? That distinction, that you could have a very strongly negative delta G to go from isocitrate to alpha-ketoglutarate, and that explains nothing about the fact that that enzyme that's doing it has a pretty high K-cat, right? It's an effective enzyme. So what we want to understand is why it's effective, even though it has no TPP and it has no shift base, it has no positive charge to sink, to resonate the carbanion.
And that's the isocitrate to alpha-ketoglutarate?
Yes. So the reaction is isocitrate, so we'll draw that first. And this is, it's not written out in great depth, but it is all mentioned in chapter 13 on the part of the text where it talks about carbon-carbon bond cleavage reactions. It does talk about this way of doing it there. We didn't mention it then, and it's done really quickly in the text, but it's pretty broadly important, so it's worth looking at now that we've actually gotten to an enzyme that uses it. So the first step of this reaction is an oxidation, which is coupled to the formation of NADH, which we like because NADH will go off and make ATP in oxidative phosphorylation. And so that generates a compound that looks like this. There's no carbon-carbon bond cleavage yet, right? But we have this guy. So the oxidation is taking place here, right? To generate that. So this compound is a beta keto acid. Because if you look at this carbon here, you consider that one's alpha, then this one would be beta, and here's the acid. So kind of like alpha, beta, to where the acid is, you have a keto group. Okay? And it turns out that the design of the substrate, the fact that you have that keto group, permits this decarboxylation to occur by cleaving this bond right here. Right? So we're cleaving here, we're going to get rid of this CO2, which is generating the thermodynamic favorability. And the fact that we have this cetyl bond O here, this oxygen can be the electron sink. And that works well enough if the enzyme groups around it are highly stabilizing to that O-, which we will presume that they are, even though we're not looking at the details of the active site of that enzyme. The next step will be to help this go by generating the O-. Right? So we could look at that and say that it's kind of like this. CO minus, CH2, CH. And then here we would have the carbanion generated. And so we could say that what's going on here is we've got electrons going into here. And we've got electrons going into here, and we've got electrons going into here. But before they can get to that oxygen, there's going to be some sort of transient carbanion on that circled carbon right there. Okay? So that's what this is. And then that will equilibrate with this. And so this is why it can happen. So this equilibration can happen, right? And then this can go into here, this can pick up a proton, and we can make the final compound, which is alpha ketoglutarate. CH2, C double bond, C minus. Right? So CO2 is gone here. And we're left with that. Questions? Okay, so again, not a reaction that you have to be able to, you need to worry about writing out from beginning to end, but rather one where, you know, I would ask you some question about it, show you some partial structure or some intermediate, and ask you to talk about what happens. Or I could show you the structure of a compound, for example. You know, this is going to work here. Notice that this reduction has to happen first so that you get the keto group. You know, if this was had another methylene on it and the carbonyl was down here, it wouldn't work, right? You couldn't have this reaction. You can't go through, you know, multiple carbon-carbon bonds to get this to happen. It has to be right adjacent. So it's a beta keto acid that makes it work. Okay. And if you look back at the pentose phosphate pathway reaction, which would be a good thing to look at, you'll see that the structure of the substrate and how the CO2 comes off of it is similar in that regard. Okay. That's the oxidative decarboxylation